Thank you.
Sir, I spoke to Motilal, sir.
Hmm.
He is visiting the state seed production farm, Neyveli.
Okay. No
He's
problem.
on the way, so we are able to connect.
Okay,
Yes,
let me start. Okay.
yes, sir.
Actually, dear my fellow scientists, actually, today I'm going to give a detailed these are references with respect to botany and genetics in ICAR-IIGR, and in between the day-to-day breeding activities, what should be done, everything will be discussed. Okay. See, today we are going to see that about the genus, botanical forms in detail, breeding objectives and approaches, just only very briefly, breeding methods and modern concepts, botany and genetics and cultivated groundnut in detail, points to remember in detail, in nutshell for references and achievements. So coming to that genus, that Arachis belongs to family Fabaceae. You know what is mean Arachis is monkey-faced. Hypogaea means below soil. Monkey-faced nut below soil. Actually, it is the meaning of the Arachis hypogaea. And we have 40 chromosomes, AABB. That A genome is that the chromosomes are slightly less smaller than B genome chromosomes. And we have 7 botanical sections. Coming to actually, we have more than 40 species available. They are in diploid condition and tetraploid condition, 2n20 and 2n40. Annuals are there, perennials are there, pod forming and stoloniferous, erect and prostrate. Actually, this wild germplasm gives us immense value for biotic and abiotic stress management. And if we have any plan to go for the wild species with cultivated species, we have to use the cultivated species as a female parent. At the same time, we should go for equal ploidy level results, and also that generations will be less for our evaluation. Then coming to botanical types of in Arachis hypogaea, there are 4 forms. Hypogaea, variety hypogaea, that is Virginia runner and Virginia bunch. Peruvian runner, we don't have anything in India in this category. And this Fastigiata, bunch groundnut, medium and short duration, and also vulgaris, that is Spanish growth. Coming to in detail, the cultivated groundnut is grouped into sequentially branched and non-sequential or alternate branched. That means sequential means that flowering node will be continuous, then vegetative node, then again continuous nodes with fruiting nodes. Whereas in alternate, 2 nodes will be vegetative, 2 nodes will be productive, then like that it will be alternated. Then coming to sequentially branched, there are 2 types, erect, only erect, we don't have any spreading type. In the erect, we have Spanish and Valencia. Then coming to non-sequentially branched or alternate branched, erect and spreading. The erect is Virginia bunch or semi-spreading, medium duration, and spreading is Virginia runner, it's a long duration. Actually, up to 1970 or 75, the area under Virginia runner, Virginia bunch was more in India. But once these dams, irrigation systems are improved, crop efficiency improved, more number of crops per annum, so that sequence changed. So the bunch variety, the Spanish bunch variety, took the area much, and this Virginia runner group have gone down because wherever the bimodal rain fawn is there, pure rain-fed crop, this Virginia runner and Virginia bunch are grown in southern states. This wild species also gives us very valuable genetic respiratory. If you take for intense branching, hairy leaves and intense branching, deep taproot system, profuse flowering in velocity carpa, and concentration of nodes at the basal. Then for forage, forage, marginata and monticola, good fertility, thin shell. So coming to this is very important, the botanical group. The procumbent 1 and procumbent 2 need not worry. The decumbent 1 and 2, these are Virginia spreading. Here, the main stem will be shorter than the primaries, and they are very dark green leaves and inverted shaped leaves, this and that. And at the same time, decumbent 3, this is Virginia bunch, that is medium duration. It is also more or less, it may be confused with our bunch types, in profuse bunch types, the recent release varieties. And this erect is Spanish bunch and Valencia. They are short duration or 110 days duration, 115 days duration. Coming to the flowering. See, in alternate, in the main stem, there will be no flower. And in the primaries and secondaries, we get 2 nodes will be vegetative and 2 nodes will be flowering. It is alternated. Whereas if you come to sequential branching, it is more main stem flowering will be there, and also sequential flowering at the basal nodes, and it will be like that only. Secondaries will be very less in this category. And we have irregulars also. We will come to later. Okay. That is, see, this is the alternate, sequential, and the irregular with flowers on main stem, with alternate primaries, and irregular without flower and main stem. These are the, because of so many crosses or other things, we get these irregulars. So the current groundnut breeding activities is concentrated for high yield, high oil content, early maturity, value addition, and tolerance to biotic and abiotic stresses. The breeding approaches will be successful. Only first we should set for the object for which we are going to breed and then cross other things. Then the proper choice of parents, the mating system employed, the quantity and quality of variability, the environmental circumstances, and finally, the breeder's keen judgment in selecting superior genotypes in segregating population, rejection rather than selection. It is most important because depending on our program, is it for quality character or for yield, oil content, these things should be taken into account. Then the rejection should be breeding methods. I'm not going to deal these things with only names, that is introduction, selection, hybridization, mutation breeding, interspecies hybriding. That introduction is being, even today it is followed. The first variety C501 in 1905 was released introduction only, and the next 1940, the TME2, it's also variety released through selection and hybridization. Then the hybridization came in 1969 onwards. That's hybridization started. That only for yield only. Now only for the last 2 decades we are going for the quality aspect. Now we have given importance, and mutation breeding and interspecies hybridization for disease resistance and breaking the these linkages, other things. We go for mutation breeding or interspecies hybridization. So these are the modern concepts.
Sir, please put it in full screen mode, sir.
Sir?
Full screen mode, sir, please. Presentation mode.
Full screen mode. Okay.
Yes, sir.
Okay.
Yeah.
Okay. Okay. Okay, Narajan?
Yes, sir. Yes, sir. Yes, sir.
Okay.
Thank you.
So out of this, the biotechnology tool has become very popular in the last 2 decades. We could release many varieties for quality improvement like high oleic acid and disease resistance. And these are the things, the GPBD4, like that. We have got very good examples from biotechnological ways also. Then why we follow mutation? That is mainly for improving 1 or 2 traits in elite or well-adapted variety and to break tight linkages between the traits. For these 2, we are very much doing mutation breeding. And if you see, even in mutation, First, we were released only mutants. Now we have mutant cultivar, mutant derivatives and cultivars. By breeding meeting, we have released so many varieties. So far, 41 varieties have been released in groundnut through mutation, and of which 16 from bark alone. Botany genetics in cultivated groundnuts in detail. See, groundnut is a herbaceous annual. We knows, and that having well-developed system and a taproot. The spreading types are usually more vigorous than the bunch types. Stem. This is also essentially, the height is essentially cultivar dependent, but also influenced by environment to a great extent. Generally, bunch types will be having thicker stems, whereas Virginia bunch and Virginia runner, Virginia spreading will be having thin stems. Internodes are short and highly condensed at the base. Stem color, this is also influenced by environment, particularly by exposure to light. Stem hairiness, it has been described as a glabrous to hirsute. There are two types, scarce and abundant. Generally, the top portion that should be observed because the lower leaves, the leaves are not persistent. Leaves. Actually, groundnut cultivars differ in characteristics for leaf color, shape, hairiness, and size. This is very important because in many cases, when you cross bunch into bunch, so that F1 identification, this comes in very hand for the selection of the hybrids. The leaf color is also important character in subspecific classification. As I told earlier, that hypogaea, that is Virginia runner and Virginia bunch will be having dark green leaves, whereas in Fastigiata and vulgaris, that is Spanish and Valencia, will be having lighter green leaf color. Leaf size also variable among cultivars. This also within cultivar or even the same plant depending on the position. The leaflets on the axis that the topmost will primary and secondary branches. The sizes are influenced by next 1 minute. I'm not getting 1 minute. Okay. Okay. The leaflets on the main axis are slightly larger than those on the primary and secondary branches. Size is influenced to some extent by environment. Leaf hairiness. Leaflets are pubescent mainly on the abaxial, that is bottom surface, and on the margins. And if you see, the difference in grades have been noticed between young and mature leaves. So coming to inflorescence, this is very important because this is going to give us the parts. That ground appears to be a cluster of flowers in the leaf axils and is a reduced monopodium. There are three or more flowers will be coming from the same leaf axil. That the number of flowers per inflorescence is cultivar dependent. The reproductive axis or the inflorescences are small replicas of the vegetative branches. If you see the Virginia group, inflorescences are very simple and expand a little during maturation. Whereas in vulgaris, a Spanish group, inflorescences compound and expand moderately. In Vastigiata or Valencia group, the inflorescences are simple and the internodes of the inflorescence may elongate to a greater length to form conspicuous branches that may terminate into a couple of leaves. The length of the reproductive axis depends on the cultivar and may exceed 10 centimeters in some cases. The absence of reproductive branches on main stem in hypogaea, that is Virginia bunch and Virginia runner, is dominant over presence of flowers in Fastigiata and vulgaris. The flowers. Actually, here also the flower, actually see that flowers are enclosed in bracts. The most common color of the standard petal is yellow, but there are 5 colors are also there. Flower color is distinct and is cultivar dependent. The standard petal has a crescent at the base from which lines are different intensities radiate. Color differences of these markings also occur between cultivars. You see that in Gemplasm, USA 86 and 68 or 74, they will be having that different crescent radiations. That is very much they seen that, and like a cheetah like that. So even that testa color will be having that cheetah like. The 5 gradations have also been reported. Normally, the markings are on the inside of the standard petal. Flowering is an indeterminate growth. Flowering commences 20 to 30 days after emergence, depending on the genotype and environment. The flowering is greatly influenced by temperature. There is a definite correlation between flowering and maturity. Actually, with a high degree of first formed flowers produce a mature fruit. Approximately, that the pattern of flowering and peak flowering depend considerably on the cultivar and environment. Again, I will be discussing in the points to remember. Other things I'll come there. Okay. Pollination. As the stigma is a clistogamous flower, the stigma is enclosed in the keel petal. Self-pollination is the rule. And in fertilization, the peg is a stalk-like structure that bears the fertilized ovule at its tip. Its growth is positive geotropic until it reaches or penetrates the soil. Then it becomes geotropic or parallel to the ground level. It is then that the ovary starts developing into a fruit. If the peg fails to contact or enter the soil, it expands at the tip and usually withers away. Color of the peg. Actually, the green color, there are light purple, dark purple, and green color. There is no pigmentation. See, if you see that white testa varieties are associated with green color pegs, that we can see that, and we can predict that if the color is green color above the soil, then we can expect that the testa color may be white. As in the case of stem pigmentation, the development of anthocyanin pigments in the peg is cultivar dependent and is very much influenced by the environment. Once that it enters inside the soil, it is only highlight because there is no sunlight. It is white color only. With the fruit, there seems to be significant cultivar differences in the proportion of pegs developing into pods. The mature pods normally contains up to 4 seeds, depending on the cultivar. Single seed may be produced in all the ovules, but the proximal aborts. Number of seeds per pod. The number of seeds per pod may range from 1 to 5 or even 6 also there in exotic this one germplasm. Single seeded pod may occur in almost all cultivar, but single seed is not a cultivar specific. The number of seeds per pod is basically cultivar dependent, though it is influenced to some extent by season and other factors also. The occurrence of 3 to 4 seeded pods was more common in bunch types than in semi-spreading and spreading cultivars, although 2 seeded pods were the most common. Large pods are dominant over small pods. Pod beak. These were important character for varietal identification. The prominence of the beak varies depending on the cultivar. The presence or absence of a beak is an excellent diagnostic character. There are 5 grades, namely absent, slightly beak. Beak is there, moderate beak, prominent beak, and very prominent beak. Pod constriction. The constriction of the pod is an interesting characteristic as it affects the developing seed. Pod constriction shows considerable variation both within and between cultivars. There are 5 grades again: nil constriction, slight constriction, moderate constriction, deep constriction, and very deep constriction. Absence or shallow constriction is dominant over deep constriction. Pod reticulation. This is a mechanical tissue that is sclerenchymatous tissue. The cell gives a reticulated or rigid appearance to the pod. It is also prominent in some cultivars. So it is also a cultivar specific and used in identification of varieties. In general, most of the ground cultivars exhibit reticulations to different degrees. They are smooth, slight, moderate, and prominent. Points to be remembered. Actually, during crossing, when the targeted traits are diversity based on pedigree and morphology and available historical data are considered while selecting suitable parents. This is very important because if we want to recycle the elite parents in breeding programs in successive crosses, it is very important. When selection is made visually, as I told earlier, the choice of pollen and ovule parent is determined by morphological characters that enable conformity, hybridity in the F1 by visual observations such as growth, branching pattern, flower color, etc. For disease resistance, nematode resistance, high oleic acid, molecular markers are available. So this method is being used in a hybrid progenies. For transfer of a trait from interspecific derivatives into a cultivar elite variety, the cultivar elite parent should be used the female parent. For some characters, the choice of parents is guided by the genetic nature of the trait. This is more important slide. Morphological traits to be used to check the hybridity of the F1s. See that I have given that ovule parent, pollen parent, and the F1 hybrid plant phenotype. If the flowering pattern, the ovule parent sequential branching, pollen parent alternate branching, the F1 will be alternate branching. Growth habit, decumbent 3, that is Virginia bunch, into decumbent 1 and 2. So again, we get the decumbent 2 and 1 like that of the pollen parent. The leaf color, green color, if it is dark green as the male parent, dark green will be coming that in the F1. Light green will be dominant. Seed size, leaf size, small into medium, we get medium leaves. Flower color, orange, garnet. In male flower, we get F1 garnet flowers. Pigmentation, stem pigmentation absent in the female parent and present in the ovule pollen parent, that F1 will be having that stem pigmentation. Pig pigmentation also like that only. The leaf shape, normal leaf is the female parent, and the pollen parent narrow leaf, that F1 will be having the narrow leaves. Testa color, tan color, red color, again we'll get red color. Reticulation on part surface, present, absent. Absent is dominant. And if you see that Virginia runner is dominant over Virginia bunch is dominant over Spanish bunch and Valencia bunch. In other words, decumbent 1 is dominant over decumbent 2, dominant over decumbent 3, over erect. Our understanding, actually, the characters, Spanish bunch, Valencia, Virginia bunch, Virginia runner, the habit, the bunch, semi-spreading, spreading, plant form, erect, decumbent 3, decumbent 1 or 2, branching, sequential, alternate, and alternate. Main stem flowering, present in Spanish bunch and Valencia, absent in Virginia bunch and Virginia runner. The duration, short, medium, long. Dormancy, mostly nil in Spanish bunch and Valencia. It is present in Virginia bunch and Virginia runner. Then why irregularities are coming in F1 when different plant forms are crossed? Because that sequential branching and main stem flowering are tightly linked. When there is a breakage in the linkage, so we get these irregularities. So then coming to dormancy in Virginia bunch and Virginia spreading, nil dormancy is seen in many wild germplasm collections. So we should not take into account that Virginia bunch and Virginia runner are having seed dormancy. Then how to fix the plant type when irregular comes, when we cross and pedigree breeding, and at the time of release of varieties, which form we should say belongs to Spanish bunch or Virginia bunch or Virginia runner. So based on first flowering, branching, duration, and plant form. This is most important. Majority of the breeders are going wrong with this, and they are giving that bunch in Virginia bunch and Virginia bunch in spreading group. So because this frustrated dormancy, because dormancy is controlled by more than 2 genes. That's why we are in a position to get small period of up to 15 days or 20 days, the frustrated dormancy in Spanish and Valencia form by crossing. In nutshell for references. See, actually inflorescence. Let's say what I spoke earlier, I have given in just that cluster of flowers in leaf axils, simple or compound, 3 or more flowers. Flower number for inflorescence is cultivar specific. Inflorescence are small replicas of the vegetative branch. Virginia bunch will be having simple little expansion. Spanish bunch will be having compound and moderate expansion. Valencia will be having simple and elongation is more. Flowers. Flower color is distinct and cultivar specific. Standard petal has a crescent with extra nervosa having radiate lines on the outer surface. Whereas in cultivar types, it will be inside the standard petal. Flowering, indeterminate. First flowering in that spreading group, Virginia spreading, 24 to 29 days from date of sowing, semi-spreading 21 to 30 days, and bunch 21 to 26 days. But there is G into E is high here. When you come to flowers, actually out of 7 flowers, 2 to 3 flowers are wasted. Only 4 to 5 forms pegs. So out of these 4 to 5, we get one or 2 mature parts and 3 waste in the form of peg, immature, tender. So actually the agronomists know why we are applying gypsum in 45 to 50 days in irrigated crop and 55 to 60 days in rain-fed crop. Because all these varieties will be having 2 peaks of flowering. So starting with 30 up to 40 days first peak, 40 to 50 days second peak. So when this gypsum is applied at that time, it automatically takes to the developing embryo in the peg tip. Actually, if you see that pegs will be having small hair-like projections, rootlets above the ovary. That is the tip. So that immediately takes that gypsum and nourishes to the developing embryo. That's why that we give that gypsum particular time. Then because in flower to pots, it is only 11 to 20%. Many people say why hybrids are not possible in groundnut, because it's a clistogamous flower, crossing is very difficult, and one flower, one seed, and indeterminate, and also we have only 11 to 20% of the pot set will be there. So it is not possible to produce a hybrid in groundnut. So we produce experimental hybrids and we go for pedigree breeding and release only varieties. Anthesis is important. 7 to 8 hours before flowering, anthesis starts. Stigma receptive will be 24 hours before flowering and 12 hours after flowering. Self-pollination rule, because clistogamous nature, 1 to 3% outcrossing reported. Fertilization will be over after 6 hours of pollination and withering starts after 6 hours from that. Genetics of traits, growth habit. Spreading is dominant over semi-spreading. Semi-spreading is dominant over bunch. Branching is dominant over non-branching. Alternative branching is dominant over sequential branching. Perennial is dominant over annual types. Late type is dominant over early. Tall is dominant over dwarf. Dark or purple is over light color stem. Hairiness on stem is dominant over non-hairy stems. Dark green foliage is dominant over light green foliage. Elliptic or oblong, elliptic leaves over elliptic leaves. Narrow leaves are dominant over normal. Mottled leaves are dominant over normal leaf. Normal leaves are dominant over crinkled leaf. Red color is dominant over absence in veins. Dark color of corolla is dominant over light. Yellow color is dominant over white. And orange white, there is not fruit. 3 colors are there, but white below soil. 1 to 4, 5 or 6 also occasionally present. See, coming to here, that's the third. This is that positive geotropic. When it enters the soil, it becomes digeotropic, parallel to the ground level. This is a catnet pot. You see that construction. This is a catnet pot. This will be available when you do crossing with wild species. You get all these types. That thin tendril-like. Then again the seed will be there. Single seedness is not cultivar specific. Seeds per pot basically cultivar specific. Beak nature 5 grades are there. Prominence is cultivar specific. Constriction of pots affects developing of the pots, shelling outbound, damaged embryos, retention of pots inside the soil. Variation occurs within and between cultivars. Fastigiata has almost nil. Constriction. Pots, large pots, small pots, absence and is dominant over presence of constriction, large over small seed size, long over short seed shape. 1 to 3 is dominant over 3 or more seeds per pot. Thin shell is dominant over thick shell. Elongated seeds over round seeds. Reticulation over smooth. Testacolor, bricklet is dominant over light tan. Red is dominant over light rose. White is dominant over red pink. Seed length is maternal influence. Quality characters. oleic acid content, 2 independent genes for high oleic acid. High protein content is dominant over low protein content. Low oil is dominant over high oil content. Then these characters are quantity characters controlled by more than 2 genes. Seed dormancy, protein and oil content, amino acid and fatty acid composition, trimeter accumulation, number of parts per plant, shelling percentage and shelling thickness, yield, resistant to diseases. Achievements. See, in 1905, the first release C501 in the 1905, we have released 252 varieties as on date up to June 2024, of which 150 are Spanish bunch, 5 are Valencia, 59 Valencia bunch, and 35 are Virginia runner. And varieties in seed chain there are 75 varieties, of which 55 are Spanish bunch, 15 are Valencia bunch, and 5 are Virginia runner. See, coming to induced mutagens, actually up to 2021, we have from India, we have released 69 oil seeds through varieties through mutation, of which groundnut is 41. Coming to Trompe alone, they have released 16 varieties in groundnut. And coming to this modern technology, that is biotechnology tool, we have released varieties, high oleic varieties, Grignard 4 and Grignard 5, and also GGHG40, interspecies derivative GPD4 from Dharwad, and highest yielding varieties from K1812 from this one, our Kadiri, Lapakshi, it is called Lapakshi, and it is from Kirsak, involved in, has got more wild germ plasm blood. That's why that we get profuse branching and also insect germination when the duration is not fixed by the farmer based on the experience. And potentially up to 7 tons they have produced in Tamil Nadu. Germplasm actions are characterized. High oil content saline tolerant genotypes are identified. QTEL for stem rot and LLS also identified. Yeah. Genome sequence also done. See, sources of resistance for many diseases have been identified. Okay, thank you for listening. For any literatures and literature with respect to oilseeds, I requested to my mail. Okay. Okay, sir.
Thank you, sir. Thank you.
Okay.
Thank you, sir. Like a basic class today. So many triads and all I have not heard first time. So first time getting to know in detail about the botany of groundnut plant and what traits are important. So now the session is open for discussion. So please unmute yourself and interact with Dr. Sir, Muralidharan sir.
Thank you, Pratap.
Good afternoon, sir.
Hi. Hello.
This is Harish Babu from Hiriyur, Karnataka, sir. Okay. Thank you very much for very informative and basic information on groundnut, sir.
Thank you.
Just a small clarification, sir, regarding the variation in pod characteristics within a single plant, sir. Why that happens? Because sometimes we observe pod shape changes within a plant and also number of seeds per pod. Occasionally, we do get some three-seeded pods in a variety which is having 2 seeds. That we have observed, and also variation in pod shape, and also sometimes reticulation also. So within a single plant, these sorts of variations are seen. What are the reasons?
See, the first formed pods you have to take into account for the identification purposes. Single seed you need not worry, because single seed is because of obstructions in the soil. We get that the ovule get absorbed from the tip, and that second kernel will becomes very big like that. That's why the first formed caps, the first formed parts should be taken into consideration for our, because that residual heterozygosity is there inside the genome. That may give some, depending on the soil and the environment, other conditions, it may give like that. But at the same time, the reticulation will not be that much. Reticulation will not. It's a character specific, in the cultivar specific. The seed number is not cultivar.
Sometimes we get the three-seeded parts, sir. Occasionally, very rarely, very rarely within the same plant, three-seeded parts and two-seeded parts would be there. One or two parts with.
Actually, it will come like that. It will come because that ovules are there inside. That ovule abortion is common in plants also. So we get that three-seeded pods. In bunch variety, we get many things. If, for example, if you see sesame, the till crop, that single capsule plants, you get three capsules in higher nodes due to environment, other things. Like that, here also that genetic expression will be there. That expressivity will be there. That for that there is no reason at all. That's only developing character because of the environment available in that part zone.
Thank you, sir. Thank you, sir.
This is Dr. Srikant from ICAR Center. May I ask?
Hello?
Yes, sir. Whether my voice is getting, sir?
I'm getting.
Yes, sir. Thank you. Sir, very good presentation. Actually, just about your last slide, that Trombay TG line, that TG 89 as well as 90 performing very well. Its potential goes up to 7.5 ton per
Yes.
hectare. But actually, there is that lines, they are susceptible and showing more susceptibility for both ELS, LLS, Rust, and others also. But in spite of that, also it's a very good one and giving yield. So my question is that, sir, how actually if we go again for to propose that line, but no one will accept. Plant type is also important one, and that due to disease, it will not accept. What to do, sir? Because the genotype is good and in ICRIP also it perform very well, sir. But that disease problem is there, leaf spot particularly, sir. More on that line.
Actually, this should be decided by the ICRIPS, this one seed, that is varietal identification committee only, because so much of disease means then how to release? Tell me. When there is so much of disease incidents, then how we can release? Tell me. We can't release because there are some set norms in the varietal release committee. We have to follow those things. And now you can go, you can use it as a female ovule parent with some resistant lines, with leaf spot disease resistance varieties are available. Many varieties are there. For example, Labaxian, so many things are there. You can do it that way also. But is it in the for the one state or across the country that the disease incidence is more?
For our Chhattisgarh, sir, it performed well, sir. As in your slide also you mentioned, 4 ton to it goes up to maximum 7 ton, and in ICRIP also it performed very well, 3.5 ton. And this is actually in Kharif season we have evaluated Kharif as well as summer, both season. Kharif season disease recorded, summer season as compared less disease. It was only LLF.
Actually, there are different zones are there in groundnut. This one
For our
release.
state, I'm concerned only for Chhattisgarh state, sir. So whether should I go to propose for that, because this is very good one, large seeded. It is like almond type, that groundnut seed. But
there are 3 seasons are there. In which season you get more disease, or in some seasons it may not be there. No? So we recommend for that season.
Yes, sir. For summer we can recommend. Yeah, this is.
That's all.
Yes, sir. Because
and also if it comes in some other zone, you release for that zone also through ICRIPS Central Varietal Committee.
Yes, sir. Yes, sir. Thank you, sir.
Dr. Sir, good afternoon, sir.
Yes, sir, Praj.
This is Praraj. Dr. Sir, I have a peculiar observations. I see maybe 30 or 35, 40 pegs are there. But when it finally developed, it came to say 15 or 20. So what might be the reason? And the plants is growing very healthy, everything is there. We have also optimum nutrition or irrigation or everything is there. Then the plants is not getting that much about to at least half I expect. But when there are the 40 pegs I count, so towards harvest it may be 15 or less than 20. So I
Sir, you
have
are
seen that's how I
tell me,
have
tell me.
seen in Pigeon P, there are 3-4 flushes also. Here also, as you told, there are 2 flushes also. Maybe around 30 days, maybe around 50 days. And how much pod is retained for these 2 flushes, maybe first flush or second flush, and how to increase the bearing capacity or retention capacity so that at least 75% pods we can realize. Is it possible, which is blending agronomically by plant breeding approaches? Thank you, Dr. Sir.
Actually, you're an agronomist, you know very well. And actually, that up to 55 days, what are the flowers have come, that is going to give you the mature parts. So that's why we are giving that gypsum application at that time. And naturally, you'll be get that we need 36 days for this development from pollination, antecedent pollen, and that the fertilization. So you see the duration. You say that take the duration up to 60 days. That effective flowers, they are going to give more number of parts. So what we have to do? Genetically, we should see more number of flower producing genotypes. First flush and second flush like that. Then agronomy. These 2 things should go because G into E is more important here. That's why we out of 7 flowers, we are getting only 1 to 2 parts only we are getting. That is the nature of the plant type. So we should have more number of flowers in leaf axil. We should have more number of basal nodes from ground level up to 5 centimeter and more number of flowers per leaf axil and formed flowers like that we need. So that we have to take into account.
That's how I have one observation. Suppose we plant during October, November, and cold temperature continues afterwards. Do we expect a good yield for our own variety or maybe variety, subdivision variety when it matures towards February or March? Again, somewhat cold. So do we expect normal yield when you go for spring, summer plantation, planting of this particular variety, maybe Virginia or maybe Spanish ones? Is it possible to get at least 2 tons of yield when you plant in October and harvest it February?
That Sankranti, that is summer swing. That is good. Because soil temperature is also very important.
Next,
for germination, other things. Germination late, then flowering will get delayed.
And also maturation.
Less
Maturity
number
also.
of flowers. Yes.
Thank you, sir.
Thank you.
Sir, I have one question regarding the pod reticulation. So pod reticulation, when the pod reticulation is less, there is smooth surface, the bruture decline is less. So can we breed for varieties having smooth pod surface?
There are varieties. TG3, VRI3, there are so many varieties, TME3, there are many varieties are there with smooth parts. Smooth parts are very good because the soil will not adhere to the parts at time of plucking and that cleaning process will be easy. But we should see that constriction should not be there. Constriction should not be deep. If it is deep, the retention of parts will be there and shelling percentage will be reduced.
Any drawbacks associated with the smooth pod surface? Apart from if the constriction is okay, Nothing. Any preference from the farmer for reticulated pods? Because I have seen so many varieties are on reticulated.
Yes, you see, farmers want more yield.
Yes, sir.
When the traders comes, they need filling. So these two simply taken into account. If we go for more reticulation, then the thickness will be more, and you get reduced shelling.
Right.
62, 64 like that. You should get at least 70. Then only that farmers will get good margin from the traders.
One more thing regarding
Actually, your point is correct. We need little beak, little reticulation. If there is no beak at all, then that shelling is very difficult. Hand shelling will be very difficult. See, my request is you please request Dr. Anand Brigana to give a lecture on this on our mutation breeding.
Sure,
He's
sir,
ready
sure.
to take class, please.
Sure, sir. So, what's your like opinion on the bitter quality of water quality parameters?
Water quality?
Uh, like the Lepakshi GG32,
Actually, I tell
issues
you,
with water quality.
actually, the Lebakshi is a good variety. It is from Crisat material. That I don't know exact parents, but many wild genomes, wild species are involved in that production evaluation. Actually, it is a very good variety. At the same time, because duration wise, it is long. When the conducive conditions are coming, it is long duration. So that already formed parts may germinate, insect germination will be there. And many people are saying that kernels are with little bitterness. Let it definitely, maybe there because of wild genome, but it is having the stay green fodder and having more oil. It is having premium price among the traders for this variety, Apache. So the farmer should decide at what time should harvest, depending on the local environment, soil condition, irrigation, weather condition, other things he should see that. So he should limit whether it's 105 days is enough, 110 days is enough. He has to see that. So he has to see here and there harvest, picking the harvest, seeing the maximum number of mature parts. Then he should decide, okay, let us forego the immature tenders, coming tenders immature. So he should decide that one. It's a very good variety, no doubt about that.
Any other question? Participants?
Hello.
Sir.
Hello, sir.
So we have completed.
Yes, sir. I hope so.
Dr. Nataraja,
Sir.
interaction is over also.
Yes, sir. From the participant end, I have to wait for a couple of minutes. Yeah, you can go ahead, sir. You can go ahead.
Sir, I have one small queries because I used to get different opinion from different school of thoughts. Groundnut जो single plant selection for yield, particularly yield and yield related traits. For selection of yield and yield related traits, mostly quantitative traits, breeders should select in early generation like rice, wheat, or we should defer to F4, F5, progeny selection. rather than single plant selection?
You should start from F2, sir. F2 you select according to his objective. Then he should go for F2, up to F4.
But Dr. Nigam used to say most of the quantitative trades, it will take time for fixing. So we should go for progeny selection in later stage, F5 onwards or F4 onwards.
But yeah, F4, F5 is enough, sir. Not more than that. No, I think so. Because that depends on the character. That depends on the character, actually.
Yield related traits only.
For
Yield
yield,
related, that selling percentage.
for yield, other things, we up to F5, F5 is maximum. F5 is enough.
He is telling F4 and F5. But some breeder is telling no, we'll go for a F2 onwards single plant selection.
See, Nikhil said they are going for single seed descendant, they are going. In that condition, we should go for up to 8 because of that part morphology, other things we should see that. But if we go for single plants as such from F2, up to F5 is enough. If at all, F6, if you require, I say up to F5 is enough.
Ajay is here? Dr. Ajay? Dr. Narendra, Dr. Ajay?
No, sir, they have not joined.
No, Dr. Ajay joined, I think.
I saw his name.
I saw.
Thomas sir is here.
I think he
From
left.
Anandapur.
Anandapur is there, yeah. Anandapur is there, sir.
Anyhow, breeding in the starting of this session, when I clear it, there are a lot of things written in the textbook, conventional textbook for breeding procedure, breeding manual, whatever may be. But when we face the real situation in the field, breeder has to take his own decision.
Yes.
So I got a chance to interact with Dr. S.A. Patil in a formal discussion. Then he simply told how to select for groundnut breeding or select the parents. He totally discouraged for back crossing if your both parents are equally improved or have a desirable traits. Your both the parents have a good yielding or high yielding. What he is telling is need not to go for any back crossing. If you want to recover one trait from if we have selected one highly desirable parent and one is only single trait is desirable, then in such cases we have to go for 2-3 back crossing. Otherwise, if both only targeting the yield or yield improvement and both the parent are highly high yielding or they have a equal contribution for that trait, there is no need for going back crossing. Don't waste your time.
It could be we go for back crossing only for this biotic resistance only, not for yield characters, not necessary.
But sometime conventional breeder used to go for a back crossing also because they are both the parents are equally good on yield or yield related traits. Sometimes
if it is interspecific crosses, the back crossing is very important. But in intervarietal crosses, back crossing is not at all necessary, but depend on the character for which he is going to breed.
Okay, if there is no questions for clarification or nothing, then Dr. Nataraja, you just express your gratitude to sir and
thank you, sir.
extend formal vote of thanks because sir will be always with us. So anybody, you can share the number of Dr. Muralidharan to the group. If anybody is having any question.
I have given the last slide I have given. Last
So
I have given.
they can have the clarification.
In the chat box, several people have asked for PPT slides, but that again depends on Muralidharan sir's approval.
At any time, they can call me through email.
Right, sir.
See,
Right, sir.
actually, I'm having collections, other things I want to share because it should go to the breeding community, young scientists. Nah? Anytime they can contact through email. I'm ready to give them the literatures, other things what I have collected as far. There are told statistics, not only for groundnut, for all other oils also.
Okay. So, with the consent of Dr. Muralidharan, you can share, or they can personally write or contact, share to have this. But sir, my primary concern is that you are there in Tamil Nadu, and Tamil Nadu, one of the traditional state and dual monsoon state for groundnut. And we have to have at least 5 lakh hectare, 5 to 6 lakh hectare. By this season, we are trying to have something more, but of course, Kharif definitely will go for a big campaign. And 24, 25, 26.
There are one request at this point. Actually, you can conduct a small meeting at Coimbatore University by inviting this policy makers of that Tamil Nadu department, and you can have a small one-day this one brainstorming session. How to that the varieties are there, good state for improvement, for area expansion, and to get more yield. You can try that because you have at your level you can do that. Because university is very difficult because Virudhachalam people from there they cannot command at Coimbatore. So you can have request a vice chancellor to have a brainstorming session at either Virudhachalam or Coimbatore. This one our agriculture department, extension department people, and we can say what are the this one pipeline varieties and good varieties, and what are the this one and yield improvement can be done within no time. Area could be increased.
So
Like
this you are suggesting by this ongoing summer season or before Kharif season, May, June?
That depends on your this one schedule of work, other things. No,
Because you just let us know when it is appropriate. Then we'll adjust our schedule.
Actually,
in March, April, or May. So, Dr. Notaraja, please note down. We'll write. Of course, we have to involve SAR also.
Yes, sir. Yes, sir.
We'll involve SAR, and then
March will be good. March and April first week.
April first week, because March will be busy for workshop.
Yes.
18, 19, 20.
April first fortnight will be good.
By end of March, no, that ending of financial year, we all will be busy.
Yes.
So maybe first fortnight of April.
Yes.
So we'll have a brainstorming meeting. Vidyachalam group will be in forefront. Sir will be there to assist us.
Yes.
With the guidance of Sir and Vidyachalam, we'll invite and check out a schedule program. Accordingly, maybe half day or 1 day brainstorming session.
1 day will be 1 day is a must.
Yes. And they have a society, that journal society, and I am a live member there. I can request them also.
Yes, plant breeding genetics.
Plant breeding and genetics. Because the one of the mandate crop of their society.
Yes, Ravi Keshavan directly will do that. He will help you.
Yes, yes. Now I'm a life member of that society.
Yes, yes, yes.
So on that behalf, I can request. Even Manivandan is there, no?
Yes, Manivan is there.
So definitely we'll try our level best, but we need to increase the area in Tamil Nadu because there are two major driving force behind of it is very high productivity. Second is equally important both the season, rainy as well as Rabi summer.
Yes.
Both the season having equal area, equal productivity. And one season, if we can supply the sufficient seed, that seed will be rotated at least 3, 4, 5 seasons.
Definitely.
So that is a good venture for us. That will be a very good venture for us, and that will help to boost the groundnut area and productivity in the state.
Yes.
Minimum effort, maximum output. So certainly we will do, sir.
Yes,
With the
sir.
help of you and TNU and our ACRIP center.
Yes.
Definitely we'll have a brainstorming meeting there in first week of April. Because first week of April will be little bit free because there won't be any government activities because of
Yes.
budget or fund. So we'll have that. Thank you.
Thank you, sir.
Dr.
Thank
Natarajan.
you. Thank you, sir. Thank you, Muralidharan sir, for
Thank you.
the short notice you agreed for this presentation, and also the later we also had to change the scheduling also because Madam Vasanti Madam has some other assignment today. So I requested Muralidharan sir to step in and take this available slot. He readily agreed, and actually he's in touch with us continuously for all the corrections, improvements, and sir is also very actively in contact with us. And I thank Muralidharan sir for this brief and very good uh, uh, talk on the traits that we want to target and what are the botanical characters that we one should keep in mind while breeding for groundnut breeding program. So thank you, sir, and we look forward to have a fruitful discussion again. And I thank all the participants from the ICAR-IIGR and ICAR-IIGR Junagadh and all the students who have joined from different universities. I thank everyone, and we look forward for the second another presentation will be on 29th from R.P. Vasanti ma'am. So we'll
That's
meet again.
that's on
Sir.
a lighter mode. It is now no longer DGR. It is IIGR.
Yes, sir. Yes, sir.
Thank you.
Thank you, sir. Thank you, sir.
Thank you.
And yes, sir. Thank you, sir. And we look forward to interact again.
Narayan, thanks,
Sir.
sir. Actually, one request is you please contact our Anand Bediginar for one
Sure,
lecture.
Sure.
Then number two, you can forward this PowerPoint to all our breeders and participants. Okay.
Sure, sir. Sure, sir. Sure.
Thank you.
Thank you, sir.
Dr. Natarajan.
Sir.
You can make some your forthcoming ACRIP workshop announcement.
Yes,
bit.
sir. Yes, sir. So we have received a confirmation from the council to hold our workshop AGM 2025 in Udaipur, MPU at Udaipur. Pratap Singh sir is here. He is a regular scientist at Udaipur Center. So we'll be in touch with you all and with all of us to successfully conduct this AGM 2025. And I have few more announcements to make. The AGM 2025, we have requested the new technical program from all the scientists who want to propose a new trial in agronomy, breeding, or entomology and pathology. That format I have shared for entomology and pathology and breeding. So I will share the Kharif crop improvement format also. We can propose new entries also for testing in Kharif 2025. And so this will be in March 18 to 20 in Udaipur. And I request all AICRP scientists who are in regular as well as voluntary centers to kindly attend this meeting without any excuse. and because that will be year end again, so we don't want to lose anyone from attending the AGM. So this year also there will be awards are kept for best performing centers, and this year also similar to the last year, each center has to present their achievements for the past year or so. So yeah, that's it, sir, from my end. And we'll be in touch with Pratap Singh, sir. Pratap Singh, sir, will circulate the brochure, Google form, and the registration fees details also soon. And we'll be in touch with all of you. Invitations will be sent from our end to all the ICAR-IIGR scientists. Thank you, sir.
The main announcement is please reserve your tickets.
Yes, sir.
Up and down tickets, take approval from your university, and accommodation. PV Singh, you please immediately check the hotels or accommodation, and immediately in mail you just send to Dr. Nataraja, and Dr. Nataraja will circulate among the all. And
okay, sir, okay. I will convey in the next week, sir.
Directly participant can book directly to the hotel, or if you have a sufficient guest house, no problem. But those who will be staying outside or in hotel, so facilitate them to book directly from their location online. That will reduce your burden. And side by side, all participants, they just quickly take the approval from their authority, competent authority, and book to and fro journey tickets, either air or ticket, train. And also you can have a additional 1 or 2 days, because Udaipur is a very good place, and March weather will be very good for sightseeing. And Dr. PV Singh also mentioned some of the tourist spot nearby, so people can plan accordingly.
Muralidharan,
Sir,
actually March 20 to March 28th.
March 18 to 20. 18, 19, 20.
18, 20.
Yes, sir.
Okay. Okay, you can write to Idayam for this one sponsorship.
Sure, sir,
I will follow. I will follow. Okay.
Right, sir. Right, sir.
Okay, this much from my side.
Thank you, sir. Thank you.
And anybody, any center is still lagging behind for giving you the report?
Sir, 5 centers, sir, for agronomy.
Oh, 5
Yes.
centers still.
Yes, sir.
Who are the centers?
Durgapura, Gwalior, Berhampur, Raigad, and one more center.
Sir, hello.
Yes,
Sir.
yes.
Sir, I will join after 10 minutes. Condolence in department will be done.
हाँ, जाइए यहाँ खत्म हो गया, तो आप जाइए, do your work and start preparation for workshop.
Yes, sir. Thank you.
Those center not yet submitted the report or data, please take it very seriously. Immediately submit, because only 1 month is left out, February, that too 28 days. Nodal officer PI has to compile, write it, conclude, then nodal officer has to print, publish it, then it will be ready for workshop. And in between we will have to review it. What are the recommendation? What will be the technology recommendation? It will be 3 years already completed, or we have to it, or we have to stop it, like that. All those things we have to take decision. And those who will not submit, already last date gone, but still you have not submitted, it will be viewed very seriously and will be reflected in your next financial release. I am clearly speaking here. It will be reflected in your. So please take it seriously and immediately send your data, those who are defaulter. Because last date, what was the last date?
The 30th.
30th January.
30th December, 31st December.
31st December. After that, January gone.
January almost passed.
It is not a very healthy situation. So this is all about. Anybody from Karnataka? Dr. Enagi here? Enagi and Mugali.
Yes, sir.
Dr. Mugali, what is the Rabi summer groundnut in Karnataka? Are you expecting expansion in area or same or reducing? Any idea?
Yeah, it will be almost same, sir, because actually even soybean farmers are taking in summer, sir. Earlier, soybean was not grown in summer, sir. But now farmers are growing soybean also in summer, sir. That's why it's almost same, sir.
Why summer farmers are taking groundnut, soybean? What could be the possible reason?
So because they have an assured market, sir. They have because they are tie up with oil industries. We have an Tina company around here. So they will procure seeds right on the farmer's field, sir. Since and harvesting is also easy, sir. It is not so labor intensive as groundnut, sir. So they prefer to go for soybean. However, we have tried, sir, through training and frontline demonstrations, as you have allotted 100, we'll try to again sensitize them, sir. Because of cost of cultivation, sir, they are moving to soybean.
That is okay, but that productivity and price. What is the productivity of soybean crop per hectare in your state, and what is the price of soybean? That is my first flag point. Second is, yes, it is harvesting is easy through harvesting through machine. Groundnut we require a little bit more, but if we go for a mechanization in full fledged like Gujarat, I think it is not a issue. And MSP of groundnut is quite high, and it is not that your soybean is purchased by a industry. Groundnut is equally also important. And what I got the market or this exporter group or business house, this year Kharif groundnut in Karnataka or Andhra Pradesh less production. In
Yes.
Kharif there is a 40 to 50% less production, groundnut production in Andhra Pradesh and Karnataka. And entire Karnataka and Andhra Pradesh buyers, they are purchasing groundnut from UP and MP, because UP and MP had a very high production in Kharif groundnut.
Yeah,
so
that's
demand is there. Demand is there. Groundnut demand is there. You can just cross-check it, this statement. So I don't think all these regions are working. Some other points are there. Probably seed, good quality seed, good variety, or farmers is not aware or something like that. technology oriented. You have soybean eclipse center in your university, Dharwad?
So yes, sir, we do have.
So you can discuss with your breeder.
Yes, sir.
And soybean seed is easily available in your center.
Yes, sir.
A groundnut may not be there.
Groundnut.
And that too, center produce those university produce seed of those crop which is easy to handle, easy to sell. So these are the some of the things working. So we have to break the barrier, and we have to bring back your groundnut area in the state. So Kharif again will try. You just looked into the production of now you have a certified seed hub and breeder seed hub. Both the seed hub you have in your university and seed division. So you have to just coordinate behind of seed group. They produce sufficient quantity of breeder seed and certified seed of recently released varieties of from your Dharwad center. So two are the very big mega variety, DH256 and 257. One is for Rabi summer only, and one is 256 is for both the season, and that is a dual advantage. If you produce sufficient seed for 256, that will cater the need of both the season, and farmers can easily maintain their seed rolling plan, Kharif to Rabi, Rabi to Kharif, like that.
Yes, sir.
So being a breeder or eclipse center in charge, you have to play a crucial role for motivating, coordinating, and get the things done. Because
Yes, sir.
Karnataka and Andhra Pradesh is a important state for us. And Andhra Pradesh this year, they have a short rainfall, less rainfall, around 30-40% area they could show. So they have a valid region this year, but Karnataka got sufficient rainfall.
Excess, sir.
Excess rainfall.
Excess, sir.
So there should not be any reason behind losing ground of groundnut area. So this is my submission to all the Karnataka center, 3 centers. Please plan and initiate the necessary action at your end. And of course, if you need any intervention from IIGR, definitely we'll extend all those help, like Dr. Muralidharan suggested a brainstorming session at Coimbatore. Similar way you can think over, and anything is required from the system, we'll extend all those support to regain the groundnut area in your state.
Okay.
Dr. Muralidharan, you have to say anything on Karnataka? Unmute, sir. Not audible, sir.
What do you say?
Any your expert comment on Karnataka?
Karnataka, yeah, actually, they have only maximum under rainfed area, rainfed situation.
No, now their irrigated area is coming up.
Coming up.
Yes.
Okay. But they themselves have very good varieties are there.
Yes.
Only the extension is should be strengthened.
extension is little bit lagging behind
Yes.
from all the three centers, Raichur, Dharwad as well as Hiriur.
Dharwad is better than Raichur in that
Yes,
condition.
that is there.
Because of staff strength, other things, na. Dharwad is better.
Okay, Dr. Praharaj, you want to say anything? Dr. Praharaj?
No, sir. No, sir. Okay.
Okay. Okay, so you can wrap up, Dr. Nataraj.
How to unmute, Natarajan.
Thank you, sir, for good guest talk that you gave today, and we had a good interaction also. So all the participants, I'm sure they will be benefited from the talk that you gave.
How many people have attended? How many people attended?
Sir, at the max it reached to 54. 54 was the
52.
max.
Okay.
Yes, sir. So I have recorded that this one. Zoom recording is there, and also slides are there. So we'll see, sir. We'll share with others. So with that, sir, thank you, sir. Thank you for good talk.
Thank you very much. Thank you very much.
Thank you, sir. Thank you all the participants for making this good.
I will do something for the Udaipur also for this sponsorship, other things. I will see that.
Sure, sir.
Okay.
Thank you, sir. Thank you, sir. Thank you, sir. I will contact Mr. Fadil. Thank you, sir.
Okay, thank you. Thank you. Bye. Bye.